This is chapter 6 from the belt text entitled DNA Synthesis, Mitosis, and Meiosis with an overview of cancer. So these 4 topics could actually be 4 separate chapters. There's a ton of material here. So I'd like to take you through the material systematically, and I've broken up the material into the following topics. What exactly is the structure of DNA, and how does its structure influence the way it functions? We'll also talk about the structure of chromosomes. So how is the DNA then organized into various types of chromosomes? The next major topic is DNA replication. So this is the way that genes are passed on from chromosomes to daughter cells. We'll then talk about the cell cycle and the process of mitosis. This will then lead us into a discussion on the control of the cell cycle and what happens when that cell cycle is dysregulated, which leads to mutations and ultimately to cancer. And next, then we'll talk about cancer detection and treatment, and lastly, we'll talk about the process of meiosis. So first of all, let's talk about the structure of DNA and chromosomal structure, but to do that, we should initially talk about cell structure, which you reviewed in the previous chapter, but just by way of reminder, here we have a bacterial cell on the left and a eukaryotic cell on the right. We can see that the DNA in a prokaryotic cell, a bacterial cell, is mostly just this globular form. If you were to actually stretch that out, it would be one huge circular piece of DNA. For the eukaryotic cell on the right, this is actually our DNA, and for all eukaryotic cells, is organized into chromosomes as opposed to a circular DNA. So whether you're looking at bacterial DNA or chromosomal DNA like we would find in eukaryotic cells, it all looks the same in terms of it's both a double helix, and it's comprised of sugars and bases and a phosphate backbone. So here you can see that DNA is double-stranded and it has this helical structure. What is DNA actually comprised of? Well, it's made up of a sugar, which is a deoxyribose. We talked about that in the previous chapter. It's also made up of a phosphate as well as a nitrogenous base. And there's 4 bases that make up DNA: adenine, thymine, guanine, or cytosine, and sometimes we just refer to these as A, T, C, G, and C. So how do these come together? You can see that the phosphate group binds on to the deoxyribose, and the deoxyribose then attaches to the nitrogenous base. Here it's showing adenine, and it forms the structure that you see here on the right. Here are the 4 different bases. You do not need to memorize these, but I just wanted you to see that they have somewhat similar structures. Adenine and guanine have 2 cyclical structures, whereas cytosine and thymine just have 1. Now you might take for granted the fact that somebody had to determine the actual structure of DNA. We really didn't know that DNA was a double helix until the 1950s. And one of the key studies leading up to the elucidation of that structure was by a researcher named Chargaff, in which in the late 1940s he was doing a series of experiments in which he was taking from the nucleus of various types of organisms the DNA and then analyzing the base composition of that DNA. So for instance, when he looked at Homo sapiens, which are people, he was able to determine that there was 31% adenine, 31.5% thymine, 19% guanine, and approximately 18.5% cytosine. He then did this for various organisms. So here is shown Drosophila melanogaster, which is just the fruit fly, Neurospora crassa, which is a red bread mold, as well as Escherichia coli, E. coli, which is a well-known bacteria. And he observed the following trends. So what I'd like for you to do is to put the video on pause for a few minutes and analyze this table and see if you find any striking tendencies or patterns. All right, welcome back. So hopefully what you're able to see when you're analyzing this table is that the amount of adenine and thymine always roughly equaled each other, and the amount of guanine and cytosine always were roughly proportional to one another. They aren't the same, those percentages aren't the same from one species to the next, but the percent of adenine to thymine and the percent of guanine to cytosine is always approximately the same. The elucidation of the structure of DNA can largely be attributed to the work of Maurice Wilkins, Rosalind Franklin, James Watson, and Francis Crick. I'm not going to go into too much detail on the history behind how the structure of DNA was determined, as that is covered in the crash course video, which I have linked below. However, I will say that most of the structure of DNA was determined due to the work by Rosalind Franklin doing X-ray crystallography work. Unfortunately, because she was working with X-rays, she died of cancer several years later, and when the Nobel Prize was awarded to those who contributed to these studies, it was given only to Maurice Wilkins, James Watson, and Francis Crick, since the Nobel Prize is not given to those who have passed away. What Rosalind was able to show was that indeed DNA was a double helix. It was thought at the time that perhaps it was a triple helix, and she was able to disprove that. She was able to show that it did have this helical structure. She was also able to demonstrate the distance between base pairs based on Chargaff's results, which was had determined that A to T's had to be equal to each other as well as G's to C's. She was able to also hypothesize the base pairing that we now know to be true. So again, what she was able to show is that there is this phosphate backbone, which is on the outside of the structure with the sugars shown here in green, and the bases are on the inside of the double helix. These nitrogenous bases can then base pair with a second strand of DNA. And here we can see that adenines and thymines base pair with one another, forming a double hydrogen bond, whereas guanine and cytosine form a triple hydrogen bond. So that's highlighting the structure of DNA, that DNA is double-stranded and it has polarity, which we didn't talk about. There's also complementary base pairing, which is by hydrogen bonding. This is the A's bind with T's and the G's with C's. And this was all discovered by Watson and Crick, Rosalind Franklin, and Maurice Wilkins. Now let's talk about how DNA is actually organized within the chromosome. So my first question to you is, do you think that all species have the same number of chromosomes? Well, the answer is no, they do not. And it's not really even based on complexity of the organism. So here we have the karyotype, or the visualization of the chromosomes for human DNA. We have 23 pairs of chromosomes, the first 22 of which are referred to as our autosomes, and then the other 2 remaining chromosomes are referred to as our sex chromosomes. So here we have one X chromosome and one Y chromosome, so this would be a male. If it was a female, we would have had 2 X chromosomes. So for bacteria, how is their DNA organized? As I said earlier, their DNA is just one big circular DNA, but you have a lot of DNA that has to fit within a very small structure. So how is that possible? Well, we don't know too much about the organization of DNA within bacteria, but we do know that they have this systematic looping that is joined together by proteins shown here in the faint pink. So these spherical balls are representing proteins that help to organize the DNA in the bacterial cell. Now, for the human genome, this is much more complicated. So, in the haploid genome, which would be just the genome you get from either your mom or your dad, you already get 3 billion base pairs in each cell from mom or from dad. So within a single cell, you actually have a copy of all your genes from mom, and you have another copy from dad. So we actually have 6 billion base pairs in each cell. And if you were to stretch that out end to end, that would be 6.5 feet of DNA that has to fit into a cell that's less than 1 millimeter in diameter. The first way in which eukaryotic DNA is organized in the cell is that the DNA, shown here in blue, is wrapped around proteins called histones. So this whole structure is called a nucleosome, and it's made up of 8 histone proteins. Here you can see an octamer of histones, octo meaning 8. 8 histone proteins around which 147 base pairs of DNA is wound around about 1.65 times. So just this presence of the histones is able to compact the DNA by a factor of about 6. With an electron micrograph, you can actually look under a microscope and see this structure of DNA wound around the histones, and we call this structure the beads on a string morphology. So how do we continue to organize the DNA until we get this chromosomal structure? Well, if you go from the top of the slide, so again, we have our DNA, and that's wound around this octamer of histones, which is the beads on a string. Those different nucleosomes can then compact and undergo further looping until we have something called chromatin. The chromatin structures then undergo further looping until we get further condensation and ultimately form this structure that we know as the chromosome. So again, if we were able to zoom in on a chromosome, eventually we would see this chromatin structure. If we were to pull that apart further, we would see the formation of nucleosomes. If we could break that apart further, we would see that that's actually the culmination of 8 histone proteins wound around the DNA until ultimately we'd be able to see double-stranded DNA. So in conclusion, for this part of the lecture, we talked about the structure of DNA, the fact that it's double-stranded, there's complementary base pairing, and the discovery of DNA, as well as chromosomal structure and the organization of the DNA within those chromosomes, which is due to histones forming nucleosomes, which forms your chromatin, and ultimately forms your chromosomes. Thank you so much.